Assalamualaikum and hi. My name is Siti Nur Zulaika binti Muhammad Noor and my student ID is 2024664852. I'm from M3802402C and today I will be presenting on the topic of pollination and fertilization and the life cycle of plants for chapter 1 of AGR499. Let's start with pollination. Pollination is the transfer of pollen grains from the anther, the male part of a flower, to the stigma, the female part of a flower within the same species. It marks the beginning of the sexual fertilization process. These pollen grains contain the male gametes and are produced in the anthers. There are two main types of pollination. The first one is self-pollination, which occurs either within the same flower or between different flowers on the same plant. And the second one is cross-pollination, which involves the transfer of pollen between the flowers of different plants that often of different cultivars. Also, cross-pollination can increase genetic diversity. We dive into pollinators. One of the most common pollination agents is insects such as bees, moths, butterflies, and beetles. Flowers that attract these pollinators are often brightly colored, although bees cannot see the red color. White flowers are typically pollinated at night by moths. These flowers usually have a sweet scent and markings that guide insects to the center. Pollen sticks to the hairs on their bodies and gets transferred from flower to flower. The second one is pollination can also occur through wind. Wind-pollinated plants produce lightweight pollen grains that can easily travel through the air. This method doesn't rely on animals or insects but requires the pollen to be produced in large quantities to increase the chances of successful pollination. Besides insect and wind, other pollinators include vertebrates such as birds, bats, and even mice. Additionally, some unusual pollinators include water slugs and snails. These agents help plants that grow in unique environment to reproduce effectively. Next, let's look at pollen grain development, also known as microsporogenesis. Takes place in the anther of a flower, which is the male reproductive part. Inside the anther are structures called microsporangia or pollen sacs where the development begins. Within each microsporangium, there are special diploid cells known as microsporocytes or pollen mother cells. Each microsporocyte undergoes meiosis, a type of cell division that reduces the chromosome number by half. As a result, each microsporocyte produces four haploid microspores. These microspores are the early forms of pollen grains. Next, each microspore develops into a mature pollen grain through a process called microgematogenesis. During this stage, the microspore undergoes mitosis, dividing into two distinct cells, which are a generative cell and a tube cell. The generative cell will eventually divide again to form two sperm cells, while the tube cell is responsible for forming the pollen tube during fertilization. The mature pollen grain consists a tough outer wall called that enzyme, which protects the pollen during its transfers to the stigma, and inner wall known as the intine. Inside the pollen grain contains both the generative and tube cells, making it ready for pollination. Once fully developed, the anther dehisces or splits open, releasing the pollen grains. These grains can then be carried by wind, insects, or other pollinators to the stigma of compatible flower, allowing fertilization to take the place. Female gametophyte development, also known as megasporogenesis, that occurs within the ovule of the ovary, which is the female part of a flower. The process begins with a single diploid cell called the megaspore mother cell or megasporocyte that located in the nucleus tissue of the ovule. This megasporocyte undergoes meiosis, a cell division process that results in 4 haploid megaspores. Out of the 4 megaspores produced, typically only one survives and becomes functional, while the other 3 are degenerate. The surviving haploid megaspores is the one that will develop into the female gametophyte, that also called the embryo sac. This functional megaspore then undergoes 3 rounds of mitotic division, resulting in egg nuclei. These egg nuclei are organized into 7 cells within the embryo sac. 1. Egg cell, which will be fertilized by a sperm cell. 2. Synergid cells, which help guide the pollen tube to the egg. 3. Antipodal cells, whose function but still not fully understood but may support development. And 1 central cell, which contains 2 polar nuclei that will participate in double fertilization. An important feature in angiosperm is double fertilization. One sperm cell fertilizes the egg cell to form a zygote, while the other fuses 2 polar nuclei to form the endosperm, which will nourish the developing embryo. This unique process ensures efficient use of resources. Once the fertilization is complete, the ovule develops into a seed, which consists of an embryo, store food, and a protective seed coat. Meanwhile, the ovary develops into a fruit, which helps in dispersing the seeds. Now, let's look at the plant life cycle. It begins with seed germination and the growth of the embryo into a sporophyte, the diploid generation. Through meiosis, spores are produced, leading to the haploid gametophyte phase, where gametes like eggs and sperm are formed through mitosis. Fertilization of these gametes return the plant to the diploid phase, which can completing the cycle. From double fertilization, we move to germination and embryo growth, forming the sporophyte. Meiosis creates spores, which develop into gametophytes. Through mitosis, these gametophytes produce gametes, and after fertilization, the cycle may continue. Thank you so much for watching and listening to my presentation. I hope this explanation of pollination, fertilization, and the plant life cycle has been informative and helpful. Thank you. Assalamualaikum and bye-bye. Thank you, Doctor.